Now during his PhD, he was studying, he has performed a mathematical model to study brain dynamics and underlying different states of consciousness, in particular those induced by psychedelic drugs. Currently, he's working in the Paris Brain Institute, where he's studying disorders, sleeping disorders, and the neural mechanism underlying attentional lapsus. So, Ruben, it's a pleasure to have you here. Thank you very much.
Thank you, Claudia, for the invitation. It's been very nice to be here. So I will start with this presentation that is beyond pairwise interactions and exploring brain high-order interactions in neurodegeneration. So yes, I will be talking about an approach for quantifying and measuring high-order interactions that is general enough for different multivariate systems, but in particular in the brain, because it's my main interest. And here, these pictures is just a way of expressing the idea of high-order interactions in this gray zone in between different variables. So let's start by the motivations. So my main motivation comes from the ideas of Francisco Varela. He was a Chilean neuroscientist, also a theoretical biologist. And in the last paper that he published before dying, he expresses this about brain dynamics and the link with cognition. So I will read: the emergence of a unified cognitive moment relies on the coordination, a key word, of scattered mosaics of functionally specialized brain regions. So here's the idea of integration and segregation of the brain. The brain operates as a coordinated and coherent whole system, but at the same time performs specialized tasks that requires to be independent or maximally independent while preserving this collective coherence. So that's one big motivation. And also here in the background, you can see all these different images of brain activity during rest, and you will see that the brain is not active just using just one region, but it's always active, implying many regions at the same time. And also a more like grounded motivation is that the brain operates at different temporal resolutions or temporal scales and different spatial scales. For example, we can go from neurons to the whole brain, and also we can go from milliseconds at the level of spiking activity, for example, or ionic currents, to days, for example, of the process of learning. And for all these different spatio-temporal combinations, we will have different tools. For example, here we have functional magnetic resonance imaging that is here, where we can record with a very good spatial resolution the whole brain activity, but with not very good temporal resolution. And here you have an image, for example, of EEG that it falls here, where we can have not that good spatial resolution, but good temporal resolution. And here you have MEG with magnetoencephalography, with electroencephalography, that will provide you maybe a better spatio-temporal resolution. And the brain is coordinating different aspects or subsystems at all the different spatial and temporal scales. For example, you can have neurons that will impact the operation of circuits, and also maybe things that you do at your whole scale. For example, taking a drug will modify the activity of neurons, and that will imply consequences at different time scales. And well, the main motivation here, I do the analogy with monogamy and polyamory, for example, that the brain regions are not operating in a monogamous way. With that, I refer that it's not by couples that the brain system is built up. It's rather made up of high-order interactions. So here again is the picture that I was showing before. You have different resting state networks that you can obtain by functional magnetic resonance imaging. And these regions are regions that tend to be active at the same time. And you can see that there are many regions, many parts of the brain are active at the same time. So it's not just couples of brain, but a couple of brain areas, but it's more like a collection of distinct collectives of different times, of different time scales and different spatial scales as well. And here is an image of regions of the default mode network. It's a network of the brain that you can observe by resting by functional magnetic resonance. And you can see that these regions, this is the activity, and this is the time course. And you can see that the orange and the yellow are highly correlated. And also, if you look at the light blue, it's also highly correlated, but it's anti-correlated. So you can think about these three regions as a highly redundant system, for example, and that maybe the best way to describe this is as a collective whole rather than a collection of pairwise interactions. And with this idea, we are here, for example, in network neuroscience, representing the brain as a collection of nodes and pairwise edges. And what I'm talking about here is that we could represent the brain as well as a hypergraph, where instead of representing this system built up of 4 nodes as a collection of pairwise interactions, we could represent this group of 4 nodes as a whole system. And that maybe will reveal some other properties of the brain that could be hidden if we understand the brain as a monogamous system. And here, for example, is the human connectome. This is the way the brain is connected. So all these are connecting fibers in the brain. And you can see that the brain is like connected in very complex ways. Many neurons are projecting actually to different parts of the brain. And that it's some kind of a structural basis for supporting the idea of thinking the brain more like an hypergraph rather than a bivariate graph. And here again, the idea of this approach of high-order interactions is to understand the interplay between integration and segregation, coming back to the initial phrase of Francisco Varela. Okay, so high-order interactions in the brain is a relatively new field, but anyways, there have been some works since the seminal work of Rosas and Mediano in 2019, where they propose very nice metrics for measuring in complex systems in general, and I will talk about that later. And here you can see about the high-order organization of brain dynamics, a couple of papers. This one I recommended very much. In aging, how this high-order interactions changes across aging, neurodegeneration, this is the work that I will be showing here, and also in state of consciousness, for example, induced by ketamine, that I will show here as well. It's a new field. I think that deserves a lot of research, of course, but it's increasing and more people are getting interested, and part of me being here is to invite you as well to be part of this research. So what is the menu today? So first I will introduce a little bit multivariate information theory. There are the tools that I will be using. Then just a methodological, what are the methods that we are using for computing these metrics in a toolbox, in Python toolbox that we implemented that is quite efficient for this kind of metrics. And then applications to resting state fMRI data. So resting state is just asking people to go into the resonator and do nothing. Quite difficult to do just nothing. And then the applications on neurodegeneration and states of consciousness. And at the end, I just want you to invite to a small discussion about the spectrum of strategies based on orders of interactions and what are the current challenges of using this approach in neuroscience and potentially in complex systems in general. Okay, so multivariate Shannon's information theory. Everything starts with Shannon's entropy. There are many entropies. Okay. Yes.
We light up together in the MRI, but that doesn't mean they interact, right?
Okay, so at the end we will talk about that as well, because for example, we have the notion of statistical interaction that could be, for example, a significant correlation, but that doesn't imply that there's an interaction in the sense of a Hamiltonian, for example, like a mechanistic interaction. So here, one of the recent papers discusses this aspect of disentangling interactions from behavior. So one thing could be high-order behavior. That is more the approach that I take. That is like the statistical relationships that you can measure. And another problem will be if this is high-order statistical relationships are given by a high-order mechanism. So yes, in this sense, I'm using interaction more from the field of statistic interactions, and not that much as a physical or mechanistic interaction. So Shannon's entropy. So this is defined by Shannon quite a long ago in the 48. And this is the equation for entropy in case of discrete variables. This is the equation of entropy for continuous variable. This is differential entropy. And conceptually, we can think about entropy as a measure of uncertainty or unpredictability, or the compressibility of a specific code, the average level of surprise or information. Probably here you manage these tools even better than me. And this is the classical thing of a coin toss, where this is the probability of having one, for example. And when the coin is perfectly balanced, we have the maximum entropy. So every time we draw the coin, we get new information. And here's the case for a continuous variable, for example, a Gaussian. All of them, they have exactly the same average, but what we are controlling here is the standard deviation. And in the case of the Gaussian, the differential entropy is a function of the standard deviation. And you can see here that as the standard deviation grows, your certainty about the output of a random sample of X increases. And then the entropy as well increases with the standard deviation. So this is basics. And then we can use this idea to the mutual information, bivariate mutual information. So Venn diagrams, I love them. The usual way is considering this is the information of one variable, x1, the information of x2. And when we intersect them, this gray zone will be the mutual information that can be defined as the sum of the entropies of each variable minus the joint entropy of the system. So that will be taking the entropy of that, summing this, and then subtracting the joint entropy of the system. And that will give you the gray zone. You can also define the mutual information in terms of conditional entropies. That will be taking the joint entropy of these two variables, will be the system, and then subtracting the conditional entropy. So what remains after having knowledge from the other variable. And that will give you the mutual information. So these two ways of expressing mutual information, like this and like that, when you scale them to the high order world, they are not equivalent anymore, and they will give you two different measures that are the measures that we will see now. So this is the idea. We are trying to compute this intersection between the Venn diagrams, and you can do it easily in the mutual information world, in the bivariate world. And the idea is conceptually to extend to higher orders. And then when we generalize this mutual information, we obtain these two metrics, that is the total correlation and the dual total correlation. So the total correlation is more related to the first equation of the mutual information, of taking, here is the sum of the entropies of all the variables, the marginal entropies, so this, and this, summing them, and subtracting the joint entropy. And that will give you this intersection. The dual total correlation, on the opposite, is based on the conditional entropies. Again, here you have the entropy of the whole system, the joint entropy, and you subtract the conditional entropies. And that will give you the intersection, but they will differ. In the case of the total correlation, you have a double counting or triple counting, but a repeated count of the triple interaction. And in the case of the dual total correlation, you are not. And if you want more details on the definition of these metrics and how they interact, you can go to this foundational paper of Fernando Rosas and Mediano. Actually, he's Chilean, and he has been developing a lot of these measures and also some other metrics, because here, maybe you are realizing that these metrics are spatial. They don't take into account dynamics or relations in different time points. So there has been also some developments on measures like this, but apply for different time points to measure statistical causality, for example. Okay, so we have the total correlation, dual total correlation. Total correlation can be interpreted as collective constraints, and dual total correlation as shared randomness. And you can check that interpretation in more details in this paper. Then, with these metrics, they can be related in a way that produce a metric that they call the O information in the same paper. And the O information is for organizational information. And when you subtract the total correlation and the dual total correlation, that will give you a metric that can take now positive and negative values. When it's positive, it indicates that the interaction that you are seeing is dominated by redundancy. And when you have negative values, it's dominated by synergy. So it doesn't imply that the interaction as a whole thing is synergistic, but that there are some dominant quality of the interaction that is synergistic or redundant. Also, if you sum these two variables, you will have what is called the S information, that they call that is the overarching level of correlations. And in a recent paper that they put out like last week, they proposed something that is called the entropic conjugation. That is a framework that allows you to define all these metrics under the same framework, that is linear combinations of entropy at different orders of interactions.
Going back to the previous transparency, when you showed the schematic thing,
it is not clear to me what it means that DTC is larger than DC.
Sorry?
What is the meaning here of that the DTC is larger than DC, that the result for is negative when you do that subtraction? It seems to me that DC has to be always larger than the DTC.
And that's the thing, that when the DTC is larger, it implies that this region is larger than the other one, and you have a synergistic interaction.
The type of diagram, what is the meaning of that in terms of the diagrammatic thing?
Yeah, in terms of that, you will have this kind of triple interaction. This. But sorry.
It has to be counted in a negative way, because if you want that the area there is bigger than the area here, the dark area has to be counted negatively somehow.
Yes, and that's because there is some information in the whole that is not in the parts. And that's the idea about synergy, that there is some information that you can access when you see the whole system as a whole that won't be accessed if you consider the system as independent parts. And that's actually the definition of the total correlation, is what is the information that you lose when you consider as the system as independent parts versus when you consider it as a whole.
But you want to bring this to this conceptual definition of graph. You have to have a negative area.
Yes. And
this negative area, which is related to what you said,
that is, yeah.
It comes from interactions, or what is the, or you cannot just represent the negative.
There are some people that represent that as a circle that it's around all this Venn diagram. And this circle appears when you have the negative information, or it just doesn't exist when you have redundant information. I don't like to put that circle because that circle won't exist when you have redundancy and will exist when you have synergy. So yeah, I prefer to not. The redundant case. Yeah. With that, yeah, you need to draw a circle that will be around the whole system. Yeah. Okay. So entropic conjugation is a new framework where you can take all these definitions together under the same framework. Okay, so let's see. So here, some videos to explain graphically synergy and redundancy. So the idea here is that you could guess what kind of system is redundant and what kind of system is synergistic. And yeah, I think like it's more or less evident that here, I wouldn't say that there's some kind of synergy, just redundancy. And here, maybe you have both. You have a little bit of synergy, a little bit of redundancy as well. And okay, so this is man-made system, nature-made system. Well, this is nature-made system as well. We made it. And this is the brain, which also shows this kind of patterns of collective coherence and also some kind of low-order segregation. Okay, so now I will pass to the methods. I will check the time. Okay. To the methods. So we're using here a method based on PyTorch to compute these metrics. So our first approach is to use what is called the Gaussian copulas. You can compute these metrics also with non-parametric measures like nearest neighbors, but they are quite expensive computationally. So one approach is to use the Gaussian copula approach that allows you to transform your data. Of course, you will be making a reduction on your data, but allows you to transform your data into a Gaussian space, preserving the interactions. That is called the copula. And with that, then you can use the Gaussian expressions for multivariate entropy. So this is the equation for differential entropy for a multivariate Gaussian. So once you transform your data into your marginal Gaussian variables, you can use this approach and optimize quite a lot the metrics. And in this paper, they show that the mutual information is copula entropy. So for example, if you have two variables and they are independent, they have the circular interaction, the copula will look like this, like a uniform copula. So if you have a uniform copula, that means maximum entropy in the copula space, you have minimum mutual information. Here you have these variables that they have some correlation, so they have mutual information. And if you go to the copula space, you see that the entropy of this copula space is non-uniform, so it's reduced. And then you can see this relationship between the copula space and the original data space. And the idea is you start with this data, for example, where the marginals are skewed, for example. You have this kind of joint probability distribution. You have the copula, and from the copula, then you obtain Gaussian marginals and a different joint probability distribution that can be characterized by the covariance matrix that is here. And for more applications of this in neuroscience, you can check this paper of Ince, where it's a very extensive explanation of the approach and how it can be used in different aspects of neuroscience. Okay, so with this, the idea is that once you transform your data into a Gaussian space, you can just take the covariance matrix and you compute just once the covariance matrix, and then you will be computing all the different interactions. For example, here in gray, we have an interaction of 1, 2, 3, 4. 4 variables at the same time. Here we have another set of 4 variables, and here we also have another set of 4 variables that are different. So we don't need to compute the covariance matrix for all this set of 4 variables. We just compute for the whole system, and then we use submatrices that will be just indexing of these matrices, and this can be optimized by batch processing and PyTorch, and that creates a huge advantage on the computation of these metrics. The problem of this is when you go into the high order world is that the number of elements, of pairwise elements plus high order interactions scales like that. So if you go to a system that is, for example, 30, the number of high order interactions go to 10 to the 9. So it's very, the exhaustive approach is very unfeasible computationally, and up to date, like going beyond 20, 25 was very hard if you don't have like a huge high performance computing approach. And the different solutions, the exhaustive that you can go up here, random sampling that could be good, but at the same time, maybe you will be overlooking relevant higher interactions. And then these two approaches that are simulated annealing and greedy search that are implemented in the toolbox that I'm here telling you. So here's the QR code. If you want to use the toolbox, it's pip installable. So you can do just pip install toy and you can use it. I did it with my colleague and flatmate. And this is computational time for different orders of interactions when you have a system of 30 variables. And these are different toolbox, and this toolbox explode the computational time at very low orders of interactions. And with our toolbox, we can go in less than 1 hour to the whole space of high order interactions for a system of 30 variables. And here are our extrapolations with these two methods, for example, are non-parametric methods, are super computational exhaustive. So even when with the Gaussian copula approach, maybe we are doing some reductions of the data, we can explore the whole space when before it was not possible. And then another approximation is the greedy approach, that you can use that when the system is too big, for example, a system of 100 or 1,000 variables, so you cannot do the exhaustive approach. And the greedy approach consists, for example, that you start from a pair, and from this pair, you build up all the possible triplets that use this pair as a base. So here you have light blue and green, and you add all the other variables to this light blue and green. And then, for example, if you want to minimize the O information, because the O information will give you synergy, then you select the triplet that maximizes the synergy. And then you select that and go to the next order. So now we have this light blue, green, and orange, and we build up all the triplets that have this, all the tetraplets that have this triplet as a base of construction. Again, you compute the O information and you take the tetraplet that gives you the minimum O information, and you proceed like this into higher order. Now, if we do that for just one pair, maybe we won't reach a good solution, but if we do it across all different pairs, we are amplifying the exploration of the combinatorial space. And for example, here, there's an example with fMRI data. It's a system of 200 variables, and we are in red maximizing the redundancy, the O info when it's positive, and minimizing the synergy, two different optimizations. We can see that at all order of interactions, synergy is growing, redundancy is growing, I'm sorry. And for the case of synergy, you can see that the synergy goes, and then up to some order of interactions, you cannot find synergy anymore. So that shows that there are some synergies in the brain, but they are confined to specific orders of interaction. Okay, so applications in resting state, we'll have some water. So just, this is an image of a resonator, someone going to resting state recordings. Let me see. Okay. Okay, so resting state functional magnetic resonance. So as I said, resting state is just doing nothing inside the resonator for 7 to 10 minutes usually. And what we found is that when you take this data, you can do what is called parcellation. Parcellation is just coarse graining of the brain activity. So you start with a resolution of 1 millimeter, for example, and you impose some map of brain regions, and you can define regions. And this is the title here. So you can define 200 regions, 100, 90, 68. And all of these are commonly used in neuroscience. And for all these parcellations, you find the same phenomena. Redundancy is growing with the order of interaction, and synergy has this kind of behavior that has a minimum and then crosses O information 0, showing you that at up to some order of interactions, you cannot find synergies anymore. And here are just summaries of these metrics, like the order of maximum, the level of maximum synergy depends on the coarse graining. That also the order of maximum synergy that will be here also depends with the coarse graining. The cutoff as well depends on the coarse graining, but if you take the ratio of them, the dependency is not that clear, even though there are significant differences between the different parcellations. So this is just for setting the ground that there are some synergies in brain activity. And actually, if you go to this paper of Barley, he also showed with a different approach. In this case, he uses simulated annealing that also there are some synergies up to a specific order, and then he cannot find these synergies anymore. So with a different approach, different research, they also find this confinement of synergies to lower to mid orders of interactions. Okay, here another application. This is a work that I did during my first postdoc, analyzing data from Alzheimer's and frontotemporal dementia. There are two different neurodegenerative diseases. In this case, the proposal is that there is some kind of landscape of the connectivity disorders that will impact brain networks in different neurodegenerative diseases. One idea, for example, is that you have just a local propagation of some protein, could be, or some molecule that will impact the health of the brain regions. Another idea will be that there's some damage in a specific network that propagates through other modules, and that some other nodes will compensate this damage that is propagated. There are some also other ways of thinking about how damage in the brain or some accumulation of proteins could propagate and impact in a network level. And in this table specifically, I'm showing Alzheimer's disease and frontotemporal dementia in terms of structural changes and functional changes. And you can see that there are some networks, like in functional connectivity terms, the default mode network and salience network, that they exhibit some changes, and also at the level of structure. So you have both structural and functional changes in brain networks. Yes, there's some relation, but there are some other functional changes that you cannot explain simply by the connectivity. And there could be also, like the other way around, that there are some functional changes in the network that could, by compensation or some other processes, impact at the end at the structural level as well. So in this work, we work with fMRI data and also with high-density EEG. It's data collected from different countries by this multi-partner consortium, and it's data from Latin America only, from Chile, Colombia, and Argentina. And the data look like this, in this case fMRI, in this case source-localized EEG. So the idea here is to do an optimization that will tell us the networks that best characterize the effect. So for that, I will compute the effect size, the Cohen's effect size, at each order of interaction. So before I show you the greedy approach for maximizing the O information, now what I want to do is to maximize the effect size. So I want to find the high order interactions that maximize the difference between control condition, healthy condition, and a pathological condition. And I can do that at each order of interaction. And you can see that there are relatively high effect sizes. Here's for fMRI and here for EEG at different frequency bands. I filter the EEG at different frequency bands, and then I compute these interactions. there is a larger effect size for both diseases at the negative. So that means that higher order interactions that were at some level in the control condition are getting disconnected in higher order terms, are getting reduced. And then at each order of interaction, we can take the higher order interaction that maximizes the effect size, so the interaction that is getting more connected, and the higher order interaction that is getting more disconnected in these statistical terms, and train a classifier to discriminate between conditions. And we can see that we can reach a very high level of discrimination with this is the area under the ROC curve. Then our idea was to show this in the brain. And here you have the most decorrelated networks in fMRI, in Alzheimer's and frontotemporal dementia. And you can see that in Alzheimer's, there are some cingulate regions, frontal regions, and also temporal regions involved. And in frontotemporal dementia, you can see that there is a little bit more extended in the brain. The spatial extension of the damage is higher. And you can also see some cingulate regions, frontal regions, and temporal regions. And this is by fMRI, and by EEG, you can also see some other regions. In this case, less regions are involved in the case of EEG, and that tells you that maybe fMRI and EEG reveal different disrupted brain high-order interactions. And that took us to the idea of integrating both EEG and fMRI in a multimodal classifier. So we perform feature selection here. So we couple the features from EEG and the features from fMRI, high-order features, and we perform feature selection, and for each set of features, we train a classifier, a cross-validated classifier, until we reach a saturating point. And this saturating point was our number of features that we use for Alzheimer's and for frontotemporal dementia. This is just the output of the classification. And what is important, what I want to show here, is that more than 60% of the features here are involved in reductions of the dual total correlation. So that implies that interactions are getting decreased by the disease, but also you have some other increases in the dual total correlation. So even when you have a disconnection, a dominance of disconnection, you also have some other things that are increasing their high-order interactions, so maybe related with the idea of compensatory effects. So are these features at each order of interactions? I have one for example, here at 9. At 9, I have one high-order interaction of 9 variables that maximizes the difference between the two conditions. And also I have one at 9 that also maximizes the difference, but in the opposite direction. One is the positive effect size and the other negative effect size. And those are my features. So I say, okay, 7 regions, I know the identity of these 7 regions, and I measure them in control condition and then in the Alzheimer condition. So my features are specific high-order interactions that I derived from this search algorithm. Okay, so the idea here is that dementias and neurodegeneration are dominated by high-order hypoconnectivity, but there's also some hyperconnectivity. And when you look at the modalities, fMRI and the different bands of the EEG, you can see that there are different and specific contributions of bands and also of modalities to the high-order interactions. And if you look at the order of interaction of the features, frontotemporal dementia has a tendency of including more and larger high-order interactions than in Alzheimer's. And that is also related to the level of damage that you can find in the brain of people with frontotemporal dementia. Then another application is in states of consciousness or unconsciousness. And here I will show anesthesia, ketamine, and if I have time, a little bit on psychedelic drugs. So I will check the time to see how far I can go. Okay. So first, propofol anesthesia. So propofol is an anesthetic that is usually used in surgery. It induces global anesthesia, deep anesthesia. And here we will be comparing people just awake in resting state, fMRI data as well, and people under deep anesthesia on propofol. Propofol in deep anesthesia induces what is called slow waves. So it's a coordinated high amplitude and slow activity in the brain when you look at by the EEG. But in this case, is fMRI, so we cannot see these slow waves because of the temporal resolution. When we do the greedy approach for the O information again, we see that awake shows at all order of interactions more redundancy than deep anesthesia. And when we look at lower orders, again, we see these synergies confined to lower order of interactions in awake, and the deep anesthesia is reducing these synergies. Then if we try to look for the high order interaction that maximize the difference between the two conditions in order to find what are the networks or variables that best characterize the effect of the anesthetic. And we can see here, this is the area under the cover of the classifier, that we have a region where different high order interactions at different levels of high order interaction will give you maximum discriminability between the two conditions. And when we look here, we can find that the greedy gives you this type of distribution. Each subject has the two conditions, so it's a pair test. And you can see that these are the levels of O information in the awake. We have some synergistic interactions. And after deep anesthesia, there are no more synergistic interaction anymore, but the O information increases and everything becomes redundant. Here as well, you have some interactions. This is with the simulated annealing. So another optimization approach to check if we reach more or less the same result. We can see that there was some synergies, and after the anesthesia, there are no more synergies, and the O information increases. And when you look at the whole brain, here the effect is on the other direction. You see that anesthesia is actually reducing the O information. The level of redundancy here is being reduced by anesthesia. So if you look at the whole brain, you see a reduction on redundancy, but if you look at lower order of interactions, you see that synergies are getting destroyed, and the redundancy is increasing. And when you look at all the region, at all the high order interactions that give you a performance of higher than 0.9, you see that the delta is always positive. So if you take all these features, all these high order interactions, they are all related to increases on the O information. So this is the main effect of anesthesia at these levels of interactions, is increase the O information with the potential implication of destroying synergies. Okay, this is another application on subanesthetic ketamine. So subanesthetic ketamine is not an anesthetic. It rather induces a non-ordinary state of consciousness or an altered state of consciousness with changes in perception and in mood. And also there has been some treatments with ketamine for treatment-resistant depression. And here, for example, you can see in adolescents, this is the treatment with midazolam, some normal treatment for people with depression, and this with ketamine. And after just one dose, you have an improvement on the scores related to depression. And there has been also some other results showing the efficacy of ketamine for depression and some other psychiatric disorders. So here we did more or less the same. In this case, we were working with EEG data, 16 electrodes. So this is the head of the subject. We have all these electrodes located at different parts of the brain, and we compute all the possible high order interactions, because with 16 you can do the exhaustive approach. You don't need optimizations. And when you compute the effect size in the two conditions, in saline and ketamine, you obtain that the O information in the alpha band specifically gives you the largest effect size. And here you can see that if you look at all these lines are different subjects, the O information consistently increases with ketamine. So again, we are seeing an increase on redundancy with the O information. And also if you use these high order interactions to compare against psychological tests, because you can do some psychological tests to the subjects after ketamine infusion, and they will tell you, okay, I felt like more derealization, depersonalization, I had like visual distortions. You can find here that if you compute these metrics in different frequency bands, you find high correlations and significant correlations, specifically in the O information, with derealization and specifically derealization and a little bit of amnesia. So high order interactions in this case can not only track different states, but also specific dimensions of the experience as measured by psychological tests. Okay, so this is the same, but in more detail. And then I will check the time. Okay. So here, psychedelic experience, and how can we use this again? So this is a painting of Alex Gray, a psychedelic artist in general. It's very common. Also, the band Tool uses these figures. And okay, so in this case, it's fMRI data with psilocybin, LSD, ketamine, and MDMA, not all together, but by separate in different centers. They did this recording. So this sounds like a joke, but it's not, in the sense that there are some people now, for example, that are mixing MDMA and LSD for improving therapy, because some experiences with LSD can be very challenging. And if you complement it with MDMA, maybe you could make the experience more manageable. And so, yeah, they are doing this kind of cocktails, pharmacological cocktails, to try to improve the outcome of therapeutics. Also, if you want more information about the efficacy of these drugs and what the studies that we can have, you can look at this Netflix documentary, also the book of Michael Pollan. So these are the molecules: psilocybin, MDMA, and LSD. They all work in the serotonergic system. So here is serotonin, and you can see that all these molecules, psilocybin, LSD, and MDMA, they have this structural similarity with serotonin. So they bind to the serotonergic receptors, so more or less similar mechanisms. And ketamine, as I showed, is a different mechanism. It's more related to glutamatergic system. Okay, so what is let's say, the empirical findings in neuroimaging data in neuroscience with psychedelics is that they change a little bit the hierarchical predictive coding. So the idea is that the brain is organized in a hierarchy of processing that is communicating sensory signals and also prediction errors of what we were predicting about the world. And then you have this kind of energy landscape that have specific and deep basins of attractions. The idea with psychedelics is that they will increase the entropy of the system and flattening the landscape and changing the dynamics of the hierarchy, flattening it a little bit more. You can check this review here where they express more this idea of the anarchic brain under psychedelic drugs. And if you look at this, for example, this is a very old work by now, the network of the brain under placebo and the network of the brain under psilocybin. You see that there are much connections, different colors at different modules, and you can see that now there are links going from different modules. So there's some kind of increase of inter-module correlation. And here as well, if you look in this case, what they are measuring here is entropy rate with Lempel-Siff complexity in different brain regions with psilocybin, ketamine, and LSD. And you can see that there's an increase on these metrics with the 3 different drugs. So what we know more or less by now is that the psychedelic drugs, they increase the entropy of brain activity, they disorganize brain network, and the hypothesis is that is a consequence of a flattening of the energy landscape and the brain hierarchies. And here's just for showing you that all these 3 drugs, they change the subjective experience. So here you have different aspects like geometric imagery, vivid imagery, perturbations in time, space, perturbations in the ego, etc. And with all these drugs, you have significant changes in different aspects of the experience. So what I did here, as in the other work of neurodegeneration, I looked for maximizing the effect size in positive and negative effect size. So this will be high order hyperconnectivity and this high order hypoconnectivity for the 4 drugs. And what we can see here is that psilocybin and LSD are dominated by hypoconnectivity. There are both things, hyperconnectivity and hypoconnectivity, but are more dominated by hypoconnectivity. While ketamine is more dominated by hyperconnectivity, but also shows some kind of balanced profile. And MDMA, even when the effect sizes are way smaller than in LSD and psilocybin, we also see that there is a dominance of hypoconnectivity. And if we look at the asterisk, there are just the features that maximize the effect size. You will see that there is a consistent, here's the hyperconnectivity. So all subjects consistently in this specific interaction are increasing their, in this case, it's the dual total correlation. And in the case of hypoconnectivity as well, all the subjects are decreasing significantly and consistently their dual total correlation. So this is just for showing that you can find high order interactions that are highly characteristic for different states of consciousness induced by psychedelic drugs. And then if you want to look at the brain networks, we have a way of projecting these high order interactions into low order variables that would be brain regions. And you can see that LSD and psilocybin, that are pharmacologically similar drugs, have very similar profiles in the brain in these parietal regions, also a little bit in here in cingulate regions. You can see also that there's hypoconnectivity in this, and also the pattern hyperconnectivity is also related to these cingulate regions. And yeah, and then MDMA is a little bit less intense, but also you can see hyperconnectivity in these cingulate regions as well. And ketamine gives you a different profile, which is expected because there are some different, there are like big difference in their pharmacological mechanisms. Then, an interim summary. Brain resting state activity exhibits synergistic interactions and plethora of high-order interactions. And brain high-order interactions can be disrupted by different conditions. Neurodegeneration, as we studied, is dominated by high-order hypoconnectivity, but there are some hyperconnectivity as well. Anesthesia reduces synergy and increased redundancy, which is compatible with what we see in EEG, that there are like these slow waves. There is a highly redundant activity. Ketamine increased redundancy and changes in subjective scores are associated with changes in high-order interactions. So we can track also specific dimensions of the subjective experience with these high-order interactions. And finally, the psychedelic states are associated with high-order hyper and hypoconnectivity, compatible with the idea of a wide reconfiguration of the brain networks under these psychedelic drugs. Okay, so I will be able to go to the discussion a little bit. So the idea is that we have all this combinatorial space that is huge. We can get lost on that. And the idea is to integrate this in this kind of gradient of our spectrum of strategies, where for some conditions, for example, or some research, will be very interesting to do univariate analysis and to look just a specific region. Maybe in some others, doing bivariate networks will be enough. And in some others, we will need high-order interactions, maybe for tracking specific aspects of the subjective experience. And the idea is that maybe reductionist and holistic perspective could be all integrated in a spectrum of orders of interactions and being compatible for understanding better the brain at a methodological level. Then what they ask at the beginning about the idea of interactions in this paper, what they try to disentangle high-order mechanisms and high-order behaviors. So for example, in an icing system, when you have frustration, where all the couplings between these three spins are negative, you have frustration. So this is a fully bivariate system. And if you look at the total correlation of all the pairs, it's small. And when you look at the total correlation of the whole system, it's very high. So here you have a separation between no high-order mechanisms in terms of the couplings on the Hamiltonian of the icing system and high-order behavior in terms of the total correlation. And in terms of the brain, it's a completely, I will say, empty avenue of research to disentangle what are the mechanisms that are giving rise to the high-order behavior that I'm showing, that I've just shown in this presentation. And finally, with the idea of orders of interactions, the idea will be to integrate different orders, levels, and techniques. So here we have all these different resolutions, spatio-temporal resolutions, and the different techniques that we have, that they will fall into the techniques and levels. And also we have the different order of interactions. And the idea will be to being able to integrate different orders, levels, and techniques in a solid theoretical framework to understand better the brain and complex systems in general. And with that, I will just to thank to the team that I'm working and to all the people that contribute to the different works that I presented here. Thank you.
Okay, thank you, Ruben. Thank you very much. Questions or comments? Can you turn the light on? Questions or comments? It's okay. Yep.
Yeah, I have one. I cannot see you. Okay, no, so which is the basic units, because you are talking all the time of these interactions at different levels, but in the image that you were showing, it was fMRI, no?
Yes.
Technique. So essentially, you have the voxels. These are the regions that are the ones that are correlated, or when you are doing this entropies, you refer to voxels. Which size of boxes, more or less, or is it to full regions of the brain?
Okay, so it will depend on the parcellation. That is what I showed, for example, in this work here. Here you have different parcellations. So you have the same brain, and the maximum resolution will be what you have from out of the fMRI. That is the resolution of millimeters. And then you use a coarse graining procedure that is called a parcellation that will give you 200 regions, 190 or 68 or different. So each of these regions will be a collection of voxels. Sometimes they do the average of the voxel, sometimes a weighted average according to the centroid, sometimes from the collection of voxels, they do PCA. There are different ways of going from the highest resolution of fMRI to a coarse graining version of it. So in fMRI, what we are using is brain regions, the fMRI activity of brain regions. And in the other work of EEG, we are using the information of the electrodes, no regions.
Any other question? Yes.
If you go back to the landscape idea of Freestone, have you tried to put your methods into that framework and try to, yeah. If you go back to the other slide, you look into these landscapes.
That's one?
No, go to the other one. Yes, this one.
Okay.
So now you see that's your way of looking. And then before you had this landscape idea, have you tried to merge these two frameworks or these two ways of looking to the brain? How does this connect to the landscapes, for instance? Have you tried to think about this? No,
not really. I think it's very, very interesting. And I think in this case, we don't have a principal notion of landscape because it's purely data driven, but there are some approaches that they feed maximum entropy models to fMRI data. So they take the ball signals, they binarize it. So now they have binary signals and they feed an Ising model, for example. And with that, you can compute the energy landscape because you have the energy of all the potential patterns of the state space. And also you could compute these metrics. So that would be like a principal way of linking the energy landscape of a system with the high order metrics. Yeah, I think it's very interesting, but I never tried to link it quantitatively. I mean, in terms of concepts, yes, you can show that here's a big reconfiguration and also hypoconnectivity. So that could be related maybe a high entropy, because if you have more connections and more redundancy, you have a decreased entropy. If you have higher entropy, things are getting more disorder, and then you expect variables to be more independent, so more disconnected. So conceptually, you can link it, but a formal way of linking it, I think, will be very interesting.
Yeah, Juan?
So how much data do you need to perform these calculations? So, because I think so, if you're looking just at one variable, you can explore the space quite well with a certain longitude of the time series. But if you have like this 16 higher order interaction, you need to do if I'm, correct me if I'm wrong, but you need to do the information of the whole system. So you need to explore this 16 dimension space.
Yes. So in the case of fMRI, the sample size for each subject is like 250 points, 300 points. So it's like very, very bad. In the case of EEG, you have more points. And in the approach that I'm using, that is the Gaussian coupolus approach. That is the one that I showed before. You estimate everything from the covariance matrix. So the bottleneck is the estimation of the covariance matrix. And also, there are some bias correctors for the metrics. And this bias corrector for the Gaussian approach, for example, depends on the sample size and depends on the system size. So at least there's some analytical results showing that how you can correct your metric with the sample size. and we will be constrained by the level of data that you require for estimating a covariance matrix. And when we do some tests, like numerical tests, simulating a system where we can control the synergy, and then we try to estimate, usually with 1,000 points, you are good to go to have good estimations, even for a system that is 100, for example, system of 100 variables with 1,000 points, you can estimate. And when you go into the 200, that is the case of fMRI data, you have much more bias in your estimator and less precision. Yeah.
Thank you.
Question? I have a very short one. This is related certainly to clinical aspects. So you talk about dementia, you talk about Alzheimer, epilepsy, or depression. So how would you relate this hyperactivity with and the hypoactivity with those kind of diseases? Can you already identify which one is related to a hyperactivity and which one is related to hypoactivity?
Yes, so in this case, we are looking at connectivity, and this connectivity could be given by hyperactivity or hypoactivity. The idea here is that we are seeing this dominance of hypoconnectivity, and that should be related, or we think, with the brain damage of the, for example, in Alzheimer's, you have the tau, the protein tau or beta amyloids, and this brain damage should also damage the connections, and that could imply this connectivity between brain areas. We also looked at the areas that are more involved in the hyper and hypoconnectivity, and they match pretty good what we already know about, like the structural and the functional changes in the brain. So in that sense, it was reassuring to see that the brain regions that we were finding were related also to the knowledge that we already have. In terms of cognitive aspects, there is some network that is called the salience and the allostatic network in frontotemporal dementia that is specifically affected in frontotemporal dementia, and is related to their loss of ability of self-regulated. And we also found that some regions were related to this allostatic network.
Okay. No more questions. Thank you again.
Okay. Ah, ¿eso ya lo tienen?